I want you to take a look at this fish. Okay, now this one. And finally this one. Would you believe they're all the same species? You probably would, as you've seen the title of this video. These three fish are all guppies, one of the most popular fish in the aquarium trade. They're also some of the most colorful among freshwater species, and because of the millions of color variations guppies can display, they're sometimes called rainbow fish. Not to be confused with rainbow shiners, which are entirely different. The patterns of guppies, plus their prolific breeding, also make them a perfect example to use when studying genetics and inheritance, one of my personal favorite biology topics. The study of genetics is attributed to Gregor Mendel. You may have heard of him. He experimented with pea plants in 1860, taking note of which trait combinations arose when breeding pea plants of different flower colors, pea shape, and size. Take a look at any textbook, and Mendel's pea plant experiment is the one you'll see described to explain genetic concepts. And while this experiment was incredibly important in helping Mendel illustrate his genetic laws, which will be discussed later, only hearing about this one example can get a bit boring. That's why today we'll be using guppies as an example, talking about how Mendelian genetics relate to the larger industry of guppy breeding. Guppies are kind of like dogs, in the way that there are hundreds of different breeds tailored to harvest specific traits, and we've been breeding them for a long time. There's red fish, blue fish, round fish, pointy fish, and so many others. Here's a list of just some of the guppy varieties currently being bred throughout the world. My personal favorites are Endlers and Speartails, but like with dogs, you can never go wrong with mutts. Okay, so we've got all these different types of guppies, but how do we organize them to illustrate the laws of genetics? Let's look at how tail shapes might be inherited as a demonstration. Tail shapes are phenotypes, traits you can physically see, like hair color or eye color, and are expressed based on the inherited combination of alleles from each of a fish's parents. Each specific combination of alleles denotes a specific genotype, which are the letters to a phenotype's word. AKA, a genotype codes for a specific trait, and that trait is expressed via a phenotype. You can remember the difference between phenotype and genotype like this. Pheno is physical, and geno are your genes. We can see how alleles of each parent are passed down to their offspring using a Punnett square, which looks like this, with the alleles of each parent on the outside of the square and the new combinations of those alleles sitting within the square. The guppies in this example will be a female with a round tail and a male with a fantail. In this case, the allele for a round tail will be capital R, and the allele for a fantail will be lowercase r. The capital R will be a representation of the dominant allele, aka the allele which will be expressed whenever it appears. And the lowercase r is the recessive allele, or the allele that can only be expressed if the capital R isn't present. The male fantail guppy's genotype will be 2 lowercase r's. In other words, it's homozygous recessive. Homo, meaning same, represents the fact that there are 2 of the same alleles. On the other hand, figuring out the genotype of the female can be a bit tricky. With the round tail being the dominant phenotype, it can show up whenever there's a capital R present, which means the female guppy in this case can either have a homozygous dominant genotype made up of 2 dominant alleles or capital R's, or heterozygous genotype, which is 1 dominant plus 1 recessive, or 1 capital R and 1 lowercase r. If you cross a female guppy with a genotype made up of 2 capital R's and a male guppy with a genotype made up of 2 lowercase r's, all of their offspring will have round tails because they will all inherit a capital R allele from their mom. However, if you cross the same male and a female guppy with the genotype capital R lowercase r, the tail shapes of their offspring will be a bit more varied. Their offspring have a 50% chance of having the capital R lowercase r genotype and a 50% chance of having the genotype lowercase r. In other words, half of their fry will have round tails and half will have fantails. This example with guppy tails illustrates Mendel's law of separation, which states that half of a gene coding for a specific trait comes from one parent and half comes from the other parent. The resulting allele combinations from crossing two fish with different tail shapes incorporate an allele from each parent, which leads to different phenotypes, causing each guppy to reflect either of their parents. Looking at the inheritance of one trait is simple enough, but of course it gets more complicated than this. In our next example, take the same male and female pairing, but now let's add color into the mix. Assume the female guppy has the genotype R lowercase r, making her tail round. But this time, she also has the genotype Y lowercase y, making her tail yellow. Meanwhile, the fantail male with the lowercase r genotype also has the genotype lowercase y, making his tail orange. A larger Punnett square can be used to determine the tail shapes and colors of their offspring in the same way as before. But this time, you must first pair each allele denoting tail shape with an allele denoting tail color for each of the parent fish before their genes can be combined. This illustrates Mendel's other law of genetics, the law of independent assortment, which states that alleles coding for different traits are passed down independently of each other. If each letter grouping on the outside of the square represents the half of each parent's genes which could be passed down, there are several combinations of tail shapes and color alleles which may be inherited. So on one side of the square are the allele combinations, and stick with me, R, Y, R, lowercase y, lowercase r, capital Y, and lowercase r, lowercase y for the female guppy. And on the other side of the square, there's only two allele combinations, lowercase r, lowercase y, and lowercase r, lowercase y. This is because the male in this instant is homozygous recessive for both traits we're looking at. Now all you have to do is combine the genes just like we did before, and you see how this gets complicated. From this larger Punnett square, you can determine the ratios of new fish which display different combinations of characteristics. Guppies with any combination of a R and a Y in their genotypes will have round yellow tails, and guppies without those dominant alleles will have orange fantails. And then there are the genotypes that encode for orange round tails and yellow fantails. From this square, we can expect the mating of a female guppy with a round yellow tail and a male guppy with an orange fantail will result in the offspring showcasing 4 different tail color and shape combinations, each combination making up about 25% of the new generation. Usually, squares looking at 2 genotypes are not this pretty, and I often find myself having trouble making sure I have noted the amount of each allele combination there is. But we lucked out with this one. There's one more important aspect of gene inheritance we need to go over. This aspect is very important when it comes to understanding inheritance, specifically in guppies. It's time to talk about sex-linked traits. Sex, in this case, refers to the chromosomes which establish the biological sex of an organism, the X and Y chromosomes. If you have any knowledge of genetics, you likely already know that female organisms have 2 X chromosomes and male organisms have both an X and Y chromosome. Of course, sex chromosomes aren't as simple as having one of 2 combinations, but for this video we will assume that guppies we're looking for fall into either category. Sex chromosomes are important because sometimes genes are specific to either the X or Y chromosome. Most of the time, these genes are linked to the X chromosome, meaning the Y chromosome doesn't have any genes that would interact with the coding for that particular trait. How this works is if the female parent is heterozygous for a trait specific to the X chromosome and the male has the dominant allele of that trait on its singular X chromosome, the only instance in which the recessive allele is expressed in the next generation is if the child is male. This is the reason why in humans, biological males are more likely to have conditions such as color blindness. However, for guppies, traits like color and pattern have widely been thought to be linked to the Y chromosome, meaning only males display those traits. This makes sense when you look at any female guppy versus any male guppy. I mean, one is clearly more gland up than the other. In reality, it's more complicated than males just having the color gene. For starters, there isn't just one color gene, but the specifics of where each gene for color and pattern are is well beyond the scope of this video. For now, all you need to know is that sons inherit their color from their fathers. With that in mind, let's set up a new Punnett square. This time, let's look at the mating of a standard silvery female and a male guppy with snakeskin patterning. This square is simpler than the last. We're just going back to combining 4 different alleles. What you're left with is a 50-50 ratio of male and female offspring. In this case, the gene for the snakeskin pattern is only on the Y chromosome, meaning the female parent does not have any genes that would impact the patterning. So if a new guppy has one Y chromosome, the patterning will appear. Stuff like this makes it really easy to tell who a male guppy fry's father is once its color develops. As for female fry, well, if you have more than one male guppy in a tank already, good luck to you. People who breed guppies to obtain males with specific traits use the method of housing one male with a desired trait and 2 female guppies of similar heritage to that male. This ensures a greater likelihood of that desired trait being passed down. Yes, before you ask, there's a lot of inbreeding going on when it comes to guppies. Just like with goldfish, just like with purebred dogs. Personally, I enjoy mixing guppies with all kinds of patterns, colors, and tail shapes, so each batch of fry is a surprising mix of attributes. This also keeps the immune systems of each guppy generation generally healthy. I believe guppies are one of the best examples you can use to illustrate genetics, as anyone with a proper setup, and I do mean a proper setup, can buy a pair of guppies and see for themselves how different traits are passed on. Plus, you can get fun fish out of it. Of course, many different colorful fish can be used in the place of guppies, including the aforementioned goldfish and betta fish, or if you aren't interested in fish at all, in which case, thanks for listening to me talk about them for so long, you can also see genetic concepts illustrated in other domesticated animals like cats, dogs, and farm animals. And heck, if plants are more your style, people have been selectively breeding crops for thousands of years. There's so much to the field of genetics than what is talked about in this video, so I encourage everyone watching to go out and learn more about it. I've just got a text saying one of my guppy fry forgot to pack a lunch for school, so I have to bounce, but I'll see you in the next video.